here has been asked if they're a robot recently?
Hey, I'm Eve, and this is Pint of Science. That's me. Who in their right mind gave me a microphone? Let me explain. For those who don't know, Pint of Science is a public science communication event held every year in pubs and bars all over the world. Hundreds, if not thousands, of restaurants and bars take place, hosting local researchers, academics, and doctors to try and spread a little science in a way that doesn't make you want to bash your head against a wall. Last year, I got to attend my local Pint of Science as a viewer. And this year...
So, I begin the routine with Eva. Welcome, Eva.
I have the clicker now. All right, you heard the boss. We got to move this along today because we have other people who are sharing our space with us. There are bingo cards on your table. Some of those are just things that are going to come out of my mouth, and some of them require you to fill in the blank. First one to get bingo gets a prize, and yes, I reserve the right to chuck the prize directly at your head, so you better not be wrong. Who here has been asked if they're a robot recently? Yeah, okay, a couple of us. I'm hoping it was by your computer, because if it was the TVA, I don't want to know. When your computer asked you if you were a robot, did it then ask you to find some bridges, some cars, possibly a few bicycles, and some really grainy and otherwise terrible photographs? Yeah. Now, why did your computer ask you to find bicycle? Anybody know? Exactly. Computers can't find bicycles, folks. In general, and this is a gross oversimplification, but you're going to get a lot of those because we got to move it along. Anything that comes innately to humans, dancing, singing, music, poetry, sarcasm, computers struggle with. That includes the interpretation of visual data, and specifically what we're looking at, live cell image analysis. My name is Eve Daniels. I'm an independent consultant and project manager with a master's in biomedical research, and I work in host pathogen interactions and social media analysis, and I write kids' books. I'm also available for hire. This is live cell image analysis. So's this, and this, and all of these are live cell image analysis. Live cell image analysis is a very complicated and in-depth technique that it's time-lapse microscopy. It's not complicated. If you've seen the GIF of the plant growing, you understand how this works. You take pictures over and over and over again, and you put them in a reel, and then all of a sudden, you have science. In the beginning, I said that I study host pathogen interactions, and then I immediately did not explain any of that. So, what's a host? I'll give you a hint. It's you. Or a puffin, or a pig, or a plant. Apparently I really liked the letter P when I made this slideshow. A host is anything that can get infected. Surprise, shock, you are all walking balls of bacteria. You probably already knew this. You don't need to panic. Most of them are friendly. Some of them are not. A host is anything that can be infected. Usually, we're talking about a particular part of a human. Now that can be your skin cells, your brain cells, your gut cells, your blood cells. I specifically work with a cell type called a macrophage. Now, if you were here for the talk yesterday with the lovely Esther Tomba, you may have remembered an introduction to macrophages. Macrophages are Pac-Man. They wander around and they eat pretty much everything inside of you that is not you. Unless they get confused, in which case they eat you too. Essentially, they're going to go ahead and eat any kind of invader, and they are the primary defense of your immune system. They're part of what's known as the innate immune response. They don't need to know what they're looking for. They just need to know that whatever they're eating is probably not you. That's the host part. Now for the pathogen. As I mentioned, we are all crawling with bacteria. They're in your gut, they're in your hair, they're in your skin. Don't panic. Most of those are not pathogens. In a gross oversimplification that will piss us every microbiologist out there, if it makes you sick, it's a pathogen. That's pretty much it. If it makes you sick, it is pathogenic. It is capable of causing disease, therefore it's a pathogen. So no, we don't really care about your gut microbiome. Well, we do, but that's why you eat yogurt. What we care about are the ones that are creeping around and crawling around and possibly going to make you sick and even send you into hospital. What I study is the interaction between host, you, and specifically your little Pac-Man immune cells, the macrophage, and the pathogens that are going to make you sick. Now, I focus on Mycobacterium tuberculosis, but also, there are other things that make you sick, ones that are less terrifying and dangerous, so work with those instead. They're less scary. I study host pathogen interactions using live cell image analysis. So how does this work? You start off by buying a really expensive microscope in a box. I use the Incucyte SX3 in case anyone is interested. There are other models out there, but that is the one that we have in the BSL-3 lab at Linköping University. Once you have your Incucyte set up, you're going to go ahead and pick your pathogen of interest. Now, it's important to note that live cell image analysis focuses on the cells. But how often you take the pictures, that is entirely dependent on the disease you're looking at. If you're looking at something slow growing, like Mycobacterium tuberculosis, which can hide in the human body for years, seriously, at least 4 of you could have it right now and we would have no idea. If you're looking at Mycobacterium tuberculosis, you're going to take a picture every 8 hours for anywhere from 5 to 10 days, depending on what exactly you're interested in. However, if you're looking at something faster growing, like Escherichia coli or Staphylococcus, you're going to take a picture every 20 minutes for 1, maybe 2 days maximum. Once you have your pathogen picked out, you need to figure out what you're going to sprinkle it on. Depending on the pathogen, that's going to dictate the cell type. Just like you're not going to go ahead and take foot fungus and put it on brain cells, whatever disease you have dictates the cell type that you are going to study. Now remember, I'm looking at macrophages, those little Pac-Man cells that eat everything. Once you have your cell type picked out and your disease picked out, you're going to sprinkle everything on top and you're going to pop it in your Incucyte to bake or incubate, same difference, and you're going to let everything run. This is going to go fast, so I apologize in advance for the head rush, but we're going to do a little bit of math here. You can put any kind of plate in the Incucyte. You can put something as big as a flask, which is about the size of your cell phone, and have just one sample. You can also use a 12-well plate and have 12 samples. You can use a 96-well plate and have 96 samples. Or you could have a 384-well plate with well sizes so small that you need to have the steadiest hands possible or a robot in order to fill that plate. I prefer the robot. It makes your hands cramp less. You have a 384-well plate. The Incucyte is a high throughput system. That means that it can hold 6 plates at one time. Now, we try not to be hogs. There are other researchers who I guess are doing important things. So we only try to take up 3 slots at a time. 3 slots times 384 wells means that at any given point for one experiment, you can have 1,152 distinct samples. We are not stopping. Now, you can do this in duplicates, you can do this in triplicates, you can do drug dilutions, you can do 100 of the same thing, but you still have 1,152 distinct samples that you need to image and analyze. So why not take a picture? Anytime you take a picture in the Incucyte, you are zoomed in to about 20x. Now, you are zoomed in so far that you're not seeing the whole well, even though it's that small. So, how do you make sure you see what's interesting? You take more than one picture. That's the long and short of it. We typically take 2 to 4 pictures per well. We're going to make it easy for my math and call it 3. 1,152 samples times 3 images per well is 3,456 images per run. This is time-lapse microscopy, people. We are not taking one picture. We're taking a lot. 7 days, 8 hours, 7 times 8, 56. 3,456, that means 193,538 images per experiment. You want to count that by hand? Because I sure don't. And this is where live cell image analysis gets interesting, because unlike other areas of science, which have the problem of not having enough data, we have too much. We have thousands of images. Almost 200,000 images per experiment that we have to analyze and compare and do correlation analysis and student t-tests, find outliers, eliminate variables. And I'm not doing that by hand. Thankfully, the Incucyte SX3 does have some built-in methods. Count the cells. How many do you think they are? A lot? Yeah. More importantly, more than 5. You are correct, Harry. Heads up. I missed. You'll find it later. More than 5 is correct. The actual number doesn't matter. What's more important is how long do you think it's going to take you to count those cells? A minute? 2?
More than 5.
10? Apparently none of us can catch. We will practice that later. Let's say it takes you 10 minutes to count the cells in that picture. Over 200,000 images, guys. You plan on finishing this year? That's one experiment. 1. This is a high throughput system. It means that we can process hundreds, if not thousands of drugs to find new treatments for diseases that are killing people around the world. But we cannot do that if we can't analyze the results as fast as we generate the data. Thankfully, the IncuSite has some methods to help us sort through this data. It does have some built-in filters that allow you to apply parameters for your things of interest and add a mask. Who here has ever used a face swap app? Yeah, how often does it go wrong? That's definitely all one cell there. It's definitely not 5 separate cells. I don't even know what that is. And these are apparently invisible. These are the masks that the IncuSite comes with. These are the preset parameters. You can only do so much because, guys, remember, computers can't find bicycles. Computers are generally really sucky at doing anything that comes intuitively to humans. That includes dancing, looking at things, sarcasm, and identifying cells in a picture. Human brains are pretty squishy. They run on dopamine. Computer brains run on binary. They don't need dopamine. Lucky bastards. If this is your analysis, are you going to be able to trust the results? Do you have any idea how many cells are there? No. You can't tell if they're dying, you can't tell if they're healthy, you're not even sure if that is a cell. That is definitely not a cell, that is definitely a scratch. That's irrelevant. A large portion of my master's thesis was spent staring at a computer screen and shouting at it to see this blob, not that blob. One of the reasons that we cannot actually trust computers in order to analyze visual data is something that Chris actually identified on Monday in their fabulous talk about noise. Now, typically when we think of noise, we think of audio noise. But the other part of noise is visual. There are shadows, there are aberrations, there are reflections. There are 1,001 things distracting the image from what is actually there. We can barely trust human eyes to be able to identify all of these cells. Are we really going to be surprised that a computer might struggle with it? In order to cut through the noise, we have to be able to teach a computer to think the way a human does, but without getting a migraine after 3 minutes. Introducing AI. The thing about AI is that if you would like me to explain how it works, I don't know. That is well above my pay grade. However, if you are interested in how artificial intelligence can be used to sift through visual data for more accurate results, I recommend that you look up convolutional neural networks, and then please explain it to me because I still don't understand. But I don't need to know how batteries work to know that I should plug a couple of them into my remote if I want to change channel. I don't need to understand the algorithms and the tools that power AI to realize that it is one of the most powerful machines that we have for interpreting visual data when it is applied correctly. These are all cells. It found all of them, except for that one. It found almost all of them. This came out with the most recent update of the IncuSite SX3. This is the AI confluence mask. It uses convolutional neural networking along with proprietary signals in order to see through the noise and identify the cells. Now, this is a cleaner sample than the last video because it still struggles when things get crowded. But between the convolutional neural networking that we can use in order to enhance our visual analysis and the fluorescent markers that we use green for our mycobacterium and red for our indicating cell death. We can perform accurate visual analysis at the same speed that we can conduct the experiments. Which means that instead of sitting with giant piles of data, no usable results, we can actually process through drugs 10, 20, 100, 1,000 at a time and find treatments for tuberculosis, COVID, HIV, and 100 other diseases so much faster and save thousands of lives in the process. Cell quantification. Remember that picture from earlier, trying to count all the individual cells? It can do that. Proliferation and calculating cell death. It can do that. Bacterial growth and inhibition, cell shape and morphology, identifying immunological responses, tracking nucleuses, cytoplasm, phagocytosis, and most importantly, retroactive analysis. The thing about live cell image analysis is that once you take the picture, you have your result. Unlike an ELISA or a DNA analysis, where once you test for antibodies, your sample is used up and that's all you've got. With live cell image analysis, these images are forever. And as the algorithms that we use to study and understand them advance, we can actually go back to experiments from 1, 2, 3, 5 years ago and apply new parameters. Which brings us to the human element. I know it's really hard to see. Imagine doing this through a tiny computer monitor for 8 hours and you can understand the entire year of my master's program. But I promise you, there are cells. This is why it's so important to pair the new advancements in AI with human validation. Just because we can apply the masks doesn't mean that we can totally trust them. This mask is doing exactly what I told it to. It is performing perfectly. It has identified all of the green fluorescent protein that we have in our mycobacteria. It has identified all of the macrophages. What it has not identified is these guys, because they weren't invited. Those are T cells. And if anyone knows why they're there, please tell me, because we don't know. All we do know is that in 20 to 30% of individuals whose blood we infect with Mycobacterium tuberculosis, after isolating the macrophages, we see an explosion of T cells. They weren't there during isolation. They weren't there in the initial stages. But somehow 5 days later, as the bacteria grows, we have thousands of T cells. And the mask is doing exactly what we told it to. It has found the macrophages. It has told us that the amount of bacteria didn't grow that much in this sample. Maybe the drugs worked. Or maybe this person has very aggressive T cells that just decided to invite themselves to our macrophage party without an invite. As advanced as the artificial intelligence gets when we look at these live cell image analysis, you cannot entirely trust the algorithms, ever. You have to verify, because otherwise your results are going to miss entirely new phenomenon. But that's also the amazing thing about live cell image analysis. You run an ELISA or a DNA sample and you test for certain genes or certain antibodies. You didn't test for a certain antibody and you wonder 5 years later if that antibody was there. You'll never know. The sample is gone. You tested for 6 antibodies and you have results for those, but not for the one you're interested in now. These pictures don't go anywhere. And after we saw this happen in 2 of my experiments, we went back through the last 4 years of experiments and found it in 26 different experiments that nobody had noticed, because the masks were looking for other things. They were trusting the masks to see what they were looking for and not questioning what else was happening. This is what time-lapse microscopy actually looks like in action. These are the macrophages that we have isolated from the human blood. Some of them are already red because they're dying. You see the green spots begin to grow. That is the mycobacterium trying to take over the human system. And now you see the T cells start to show up. Again, they were not invited to this party. But they are there anyways. And now, because we're using a technique like this that records everything that is happening over a progression of time, instead of just taking one sample at one time point, we've discovered an entirely new immune response that we are still in the process of classifying and have just barely scratched the surface on. So thank you.